Good morning, students. Today we'll start a new topic, that is formulation of fermentation medium. How to formulate, what is the speciality in formulation of medium, why I have given that much importance to the formulation of medium, why it is so important to formulate a medium. In general, we can prepare the medium, microbial medium, any bacterial or fungal medium, whatever organism you are growing, the microbial medium can be prepared, and it is a nutrient agar medium or potato dextrose medium or Asthana Hawker's medium, whatever it may be, you can pour into your fermenter. But it is not like that. When you are preparing the medium, special type of medium should be used. That means a proper medium should be used for the culture of microorganisms, first thing. Second one is, the medium should satisfy the nutritional requirements of the microorganisms. Whatever medium you are using, it should satisfy the nutritional requirements of the medium. Third one, when you are using the chemicals, the concentration of the chemicals or chemical nutrients should allow the growth of a specific microorganism. For each and every microorganism, its requirements are different. See here, in our family we are 5. Mother, father, brother, sisters, like that we are 5. Our family consists of 5 people, 5 persons. If mother is cooking, if she has done today breakfast, that is upma she has prepared, what you will do? One of us will say that I don't want upma, I want to eat some sandwich, or I want to eat something. Daily you are preparing upma and I don't want to eat it. అంటే ఏంటి, ఉన్న 5గురిలోనే మనకి ఒకళ్ళు ఉప్మా వద్దంటారు, ఇంకోళ్ళు అబ్బా రోజు ఉప్మానేనా, ఇవాళ నేను తినను, నాకు కూడా ఇంకేదన్నా కావాలంటారు. అంటే ఏంటి అంటే, ఉన్న 5గురిలోనే మనకి ఎన్నో వేరియేషన్స్ ఉన్నాయి, అందరం ఒకటి తినము. ఇవాళ కాకరకాయ కూర చేసింది అమ్మ, ఇంట్లో 2-3 తినరు, పిల్లలు అంటారు మాకు కాకరకాయ కూర వద్దంటే, అమ్మకి ఇంకొక వేరియేషన్, ఫాదర్ మదర్ తినడానికి కాకరకాయ కూర చేస్తే హెల్త్కి మంచిది కాబట్టి బతిమాలి మనకి ఇంత పెట్టినా మన కోసం ఆలూ ఫ్రై చేస్తుందమ్మ. అంటే ఏంటి అంటే, మనకే అన్ని వేరియేషన్స్ ఉన్నప్పుడు ఈ మైక్రో ఆర్గానిజమ్స్కి కూడా దే హ్యావ్ దేర్ వేరియేషన్స్, బికాజ్ దే ఆర్ లివింగ్ ఆర్గానిజమ్స్. ఏమి చచ్చిపోయి లేవు, అవి బతికే ఉన్నాయి కాబట్టి దే రిక్వైర్ స్పెసిఫిక్ టైప్ ఆఫ్ న్యూట్రియంట్స్ ఫర్ దేర్ గ్రోత్. సో each and every microorganism has its own special type of nutrient requirement is there. That nutrients are added. Not only that, they require these nutrients in specific concentrations, so their concentrations are also added in the medium. That means proper nutritive medium should be prepared for the growth of microorganisms, first thing. Second one, when you are preparing the proper medium, specific nutrients for the growth of, that means the nutrients should satisfy the medium should satisfy the nutritive requirements of the microorganisms. Third one, the concentration of the nutrients should satisfy the growth of a special type of microorganisms. Fourth one, the materials that are added into the fermentation medium, they should be inexpensive or they should be cheap. Cost ఎక్కువ ఉండదు, low cost. Next, when they are used in the culture medium, they should be available round the year. అంటే ఏంటి, నాకు సమ్మర్ సీజన్లో దొరుకుతాయి, వింటర్ సీజన్లో దొరకవు, లేకపోతే రైనీ సీజన్లో దొరకదు ఆ మెటీరియల్ అని ఉండడానికి లేదు. ఏ కెమికల్స్ అయితే మనం దీనిలో యూజ్ చేస్తున్నామో, ఏ రా మెటీరియల్ అయితే దానిలో యూజ్ చేస్తున్నామో, వాట్ ఎవర్ మెటీరియల్ that is used in the formulation of in the medium, nutritional medium or bacterial microbial nutritional medium, they should be cheap and they should be or inexpensive, or they should be low cost and they should be available round the year. Next one, not only that, these chemical media composition, that is the chemicals that are used in the medium, their handling, when you are adding them, the handling. Next, transporting, when you are purchasing it, the transporting. And next one is, after purchasing them, you are storing. అంటే ఇవాళ ఇట్లా రాగానే వాడేయట్లే, మనం బియ్యం బస్తా వస్తే బియ్యం బస్తాని స్టోర్ చేసి రోజు కొంచెం తీసి వాడుకుంటున్నాం, ఎంత అవసరమో అంట. అట్లనే ఒక కెమికల్ని కొన్న తర్వాత దాన్ని ట్రాన్స్పోర్ట్ చేసుకుని వచ్చిన తర్వాత దాని స్టోరేజ్ కూడా ప్రాబ్లం ఉండకూడదు, తర్వాత దాని హ్యాండ్లింగ్ కూడా ప్రాబ్లం ఉండకూడదు. ఇది సీక్వెన్స్. ట్రాన్స్పోర్టేషన్, స్టోరేజ్ అండ్ హ్యాండ్లింగ్ షుడ్ బి ఈజీ. నెక్స్ట్ వన్, if they are deteriorating, dehydrating or some deoxygenation is taking place, if the chemical is getting damaged or the chemical is deteriorating, then the that problem should be easily rectified. అంటే ఏంటి, ఒక కెమికల్ మనం ఏదైనా తెచ్చుకుంటే అది ఆక్సిడైజ్ అవుతున్నా, డీహైడ్రేట్ అవుతున్నా, లేదా హైడ్రేషన్ అవుతున్నా, ఏదైనా అవుతున్నా ఆ ప్రాబ్లమ్ని మనం ఈజీగా సాల్వ్ చేసేటట్టు దట్ ప్రాబ్లమ్ షుడ్ బి ఈజీలీ సాల్వ్డ్ ఫర్ ది కెమికల్స్. నెక్స్ట్ వన్, ఆఫ్టర్ యూజింగ్ these chemicals, whatever cheap raw materials that we are using in the fermentation medium, they should allow the required concentration of the product synthesis. అంటే ఏంటి, they should allow the synthesis of required concentration of the target product. మనం ఏ ప్రోడక్ట్ అయితే టార్గెట్ చేసి ప్రోడక్ట్ తయారు చేద్దాం అనుకుంటున్నామో ఆ సింథసిస్ ఆఫ్ ది టార్గెట్ ప్రోడక్ట్ కాన్సన్ట్రేషను మనం అనుకున్నంతే రావాలి, అది తక్కువ రావద్దు. నెక్స్ట్, when you are using these chemicals, see here, when you are using these chemicals, they should be very very carefully we have to use them, and they should not lead to any health hazards of the laborers who are working in that area. అంటే ఏంటి, ఈ కెమికల్స్ వాడడం వల్ల ఆ ఫెర్మెంటేషన్ యూనిట్లో జాబ్ చేసే పని చేసే లేబరర్స్ కానీ సైంటిస్టులకు కానీ ఎవ్వరికి పర్సనల్కి అనండి, లేబరర్స్ అదంతా ఎందుకు, ఆ ఫర్మెంటేషన్ యూనిట్లో పనిచేసే పర్సనల్కి ఎవరికి హెల్త్ హెజార్డ్స్ రాకుండా ఉండాలి. అంటే ఏంటి, దీని నుంచి వచ్చే గ్యాసెస్ కానీ దీని నుంచి వచ్చే ఏదైనా కెమికల్ స్మెల్ కానీ గ్యాసెస్ కానీ ఏవి అక్కడ పనిచేసే పర్సనల్కి ఎవరికి హెల్త్ హెజార్డ్స్ రావద్దు. అంటే ఇట్ షుడ్ సాటిస్ఫై ది హెల్త్ నీడ్స్ ఆఫ్ ది పర్సనల్ హూ ఈజ్ వర్కింగ్ ఇన్ దిస్ ఫెర్మెంటేషన్ ఏరియా. ఓకే, అంటే ఏంటి, ఈ ఫెర్మెంటేషన్కి యూజ్ చేసే మీడియము ఇట్ షుడ్ సాటిస్ఫై, దట్ ఈస్ ప్రాపర్ మీడియం షుడ్ బి ప్రిపేర్డ్ ఫర్ ది గ్రోత్ ఆఫ్ ఫర్ ది కల్చరింగ్ ఆఫ్ microorganisms or for the culture of microorganisms. Next one is it should satisfy, media chemicals should satisfy the nutritional requirements of the microorganism. Their concentration should be satisfied for the growth of specific type of microorganisms. They should be cheap or inexpensive. Next, they should be available round the year. Next one is their transport, storage and handling should be easy. If any damage is occurring to the chemical, that problem should be easily solved. Next is the chemical that is used in the nutrient medium that should help in the production, in the synthesis of the required concentration of the target product. And next one is it should not create problems in the health of the personnel who is working in the fermentation unit. Okay, these are some of the introductory words about your fermentation medium. Now, in this fermentation medium, what are essential for the fermentation medium? First one is it requires your, see here, it requires amino acids, carbon source, nitrogen source, next vitamins, next it requires mineral elements. Okay, carbon source, nitrogen source, vitamins, amino acids, mineral elements, all these are added to your nutrient medium. Now, the microorganisms can synthesize a small amount of vitamins, and those vitamins are not enough for their growth. Next, nitrogen and carbon, they can be synthesized by the cells. Why? Because carbon is regularly synthesized in the form of carbon dioxide, and nitrogen is present in the nucleotides. And not only that, mineral elements, from the food they are taking, they get the mineral elements. Next is amino acids, or 1 or 2 amino acids are already present in the cytoplasm of the cells. So, see here, all these are present, no doubt they are present in the cytoplasm of the cells, but they are in very less amounts or least amounts, so they are not enough for the production of our product. So, to increase the concentration of the product, to increase the synthesis or production of the product, see here, these are the chemicals that are added externally into the culture medium. Okay, now see here, coming to this, today we will see carbon source. When you are using inexpensive or cheap carbon source, what is there? See here, when you are using carbon source, when you are using carbon source, what is happening? When you are taking the carbon, here what happens is, in this carbon source, what is there is, you will be using cheap chemicals or cheap material that is used in the carbon source, but it should allow the production of your required target protein or target product in required concentrations. Now coming to the carbon source. The carbon source is, first one, you can use molasses. Molasses is nothing but, if you take a sugar cane, or you can take beetroots, okay, you can take beetroot or beetroot sugar or sugar cane sugar. What you will be taking is, take the sugar cane, crush it, you will get the liquid, and that sugar cane juice is taken and it is poured into a vessel where it will get boiled. See here, take sugar cane into a vessel, and in the vessel from here you boil it. When you boil this sugar cane juice, this sugar cane juice will become thick. Paakam battinattu ayipotundi. When it becomes thick, that thickness is known as molasses. This thick concentrated sugar syrup is known as molasses. This is known as first molasses. Now again and again you are boiling it, it will become still more thick. This is known as second molasses. And still more you are boiling it, it will become crystal form like black crystals. This is known as third molasses. So molasses is nothing but the sugar cane juice is taken, and that sugar cane juice which is collected is boiled. When it is boiled, it becomes a viscous fluid, thick concentrated syrup. This thick concentrated molasses syrup, first molasses syrup, is taken and it is added in your food additives. It is added as a food additive for your sweets. Not only that, it is added in the cough syrups of children. చూడండి, చిన్నపిల్లలు తాగే దగ్గు మందు ఎక్కువ చేదుగా అట్లా వగరుగా ఉండదు, తీయగా ఉంటుంది. ఎందుకంటే ఇన్ దట్ దే విల్ బి యాడింగ్ దిస్ ఫస్ట్ మొలాసిస్ షుగర్ సిరప్. సెకండ్ మొలాసిస్ షుగర్ సిరప్, ఇట్ విల్ టర్న్ బ్లాక్. సో ఇట్ ఈస్ ఆల్సో known as black tricale. This is also known as black tricale. And third one is, it transforms into crystal form. That is, black crystal forms are produced. Now the first molasses is sweet in taste. The second molasses is a little bit, its sweetness will be lowered and it will become something like bitter, that is light bitter. And the third molasses will be high bitter. It will be highly bitter and you cannot eat it. So see here, molasses is, the sugar syrup is boiled and the molasses is taken, and this molasses is, that is the molasses sweet syrup or the black tricale. Generally this black tricale is used as molasses in the fermentation unit. So that means this molasses consists of, see here, this molasses consists of 50 to 60% of carbohydrates. See here, it consists of 50 to 60% of carbohydrate, carbon source, and next it also consists of manganese, magnesium, next is potassium, calcium, phosphorus and iron. These are the mineral elements that are present in the molasses product, that is sweet syrup. Not only that, it also consists of vitamin B6 and it also consists of proteins. These are the things that are present in the molasses. Next, second one is known as malt extract. When you are using malt extract, what is malt extract? Malt is a product that is produced, that is, it helps in the production of a carbohydrate called as maltose. When you are using maltose, here the malt extract is that this is produced from the germinating seeds. When the seeds are germinating, then in germinating seeds they produce maltose, carbohydrate maltose, and that maltose is nothing but your malt extract. What you have to do is take the germinating seeds, dry them and powder them. అంటే ఏంటి, మొలకెత్తే విత్తనాలు తీసుకోండి, వాటిని డ్రై చేయండి, డ్రై చేసిన వాటిని పౌడర్ చేయండి. దెన్ దిస్ పౌడర్ వాట్ యూ హ్యావ్ టు డూ ఈజ్ మిక్స్ వాటర్, బాయిల్. you will get extract, filter the extract, and that extract is known as a malt extract. And this malt extract, it is used as an energy source for your microorganisms, and this consists of 90% carbohydrates. It consists of 90% carbohydrates, and it has hexose sugar, next disaccharide, and it also consists of trisaccharides. It has disaccharide and trisaccharides along with hexoses, that is nothing but your monosaccharides. All these are present in the malt extract. Coming to the third one. The third one is your starch. What is starch? Starch is nothing but it is a polysaccharide. No. So the starch, which is, what is the starch here we are using? The starch that is used is generally your cereal grass or generally your maize grass. అంటే ఏంటి, మొక్కజొన్నలు కట్ చేసుకున్న తర్వాత ఆ మొక్కజొన్న చేలల్లో మిగిలిపోయిన ఆ చెట్లు ఏవైతే ఉన్నాయో ఆ గడ్డి మొత్తం తీసుకొచ్చి దాన్ని చిన్న చిన్న పీసెస్ కట్ చేసి ఆ మెయిజ్ స్టార్చ్ని, మెయిజ్ ప్లాంట్ని కట్ చేసి పీసెస్ లాగా దీన్ని వాటర్లో కలిపి బాయిల్ చేసి ఆ బాయిల్ అయిన దాన్ని ఎక్స్ట్రాక్ట్ చేస్తే, అంటే ఏంటి, ఫిల్టర్ చేస్తే you will get this, see here, when you filter it you will get this starch, that is maize plant extract, and that starch consists of polysaccharide. So it should be, starch should be used for the growth of amylase producing microorganisms. See here, amylase producing microorganisms are grown in the starch because the amylase producing microorganisms, they act upon this polysaccharide and convert it into glucose and sucrose. That is, mono and disaccharides are produced with the help of amylase producing microorganisms. The polysaccharides are digested into single residues, that is a glucose molecule and a sucrose molecule are produced, and they help in turn for the growth of your microorganisms, amylase producing microorganisms. This is known as starch. This is also another type of cheap or inexpensive carbon source that is used in the fermentation medium. You can use any one depending upon your requirement. Coming to the next one, that is, we have seen these. Third one we have seen. Coming to the fourth one, sulfate waste liquor. Sulfate waste liquor. Sulfate waste liquor means it is nothing but simply you take the plants which are used for paper, the plant or the pulp, paper pulp, that is the plant pulp that is used for the synthesis or production of paper in the paper industry. After the plant pulp is taken, this pulp is used in the preparation synthesis of your paper, and once the paper is prepared, the pulp will be, that is the pulp or the extract is taken for the paper preparation synthesis, and the pulp is left over. The pulp that is left over అంటే చెరుకు రసం తీసిన తర్వాత మిగిలిపోయే ఆ చెరుకు మొత్తం ఉంటుంది చూడండి, కింద పడిపోయింది చెరుకు రసం బండి దగ్గర, అట్లా ఒక ప్లాంట్ని తీసుకొని మనం పేపర్ తయారు చేసినప్పుడు పేపర్ పల్ప్, ఈ ప్లాంట్ యొక్క పల్ప్ అక్కడ మిగిలిపోతుంది. దిస్ పేపర్ ఈజ్ నోన్ యాజ్ వేస్ట్, and this waste consists of sulfate waste because it consists of sulfur dioxide. The pulp that is used after synthesis of your paper, it consists of, that waste pulp consists of sulfur dioxide, and this is used as your carbon source because it consists of 2 to 3% carbon. It consists of 2 to 3% of carbon or carbohydrate. Now to remove this sulfur dioxide, which is a sulfate liquor, why we are calling it as liquor? Because this waste is mixed with water and it is boiled, and that is known as liquor. And this liquor consists of waste plus sulfur dioxide plus 2 to 3% of carbon, and to reduce the acidity you will be adding either calcium hydroxide or you will be adding calcium carbonate in the culture medium, so that the pH will be reduced. Not only that, you will be adding phosphorus and nitrogen so that the effect of sulfate can be reduced, and this helps in the growth of the microorganisms. It is used as a carbon source. That is the fourth type. Coming to the fifth one, you can use cellulose. What is cellulose? Cellulose is present in cellulose, hemicellulose and pectin. These are the three materials that are present in the cell wall of the plants. So, take any plant material, and each and every plant material cell consists of cellulose in its cell wall, and this cellulose is taken, and this cellulose is useful as a carbon source. Why? Because cellulose is a, all of you know that it is a polysaccharide, just like your starch. So, as cellulose is a polysaccharide which helps in the construction of the plant cell wall, so you take any plant and use it as a material, that is cellulose, and this also helps in, this consists of, as it is a polysaccharide, this is broken into glucose, fructose, sucrose, like that. Mono and disaccharides are produced by the action of the microorganisms, and these are used for the growth of the other microorganisms. So, the cellulose is also used. Next, coming to the sixth one is Y. WHY కాదు, WHEY, Y. మేడం తప్పు రాసింది అనుకోకండి, WHEY, వే అంటారు దీన్ని. వే ఈజ్ నథింగ్ బట్ ది లిక్విడ్ బై ప్రోడక్ట్ ఆఫ్ డైరీ ఇండస్ట్రీ. What is it? Simply daily in our house we will be having whey. What is it means? When you settle the curd, if you take the milk and allow it to curdling, it will get curd is formed. అంటే ఏంటి, మనం తోడు పెడితే పాలు పెరుగులాగా తోడుకుంది. On that curd, the superficial layer of curd consists of small liquid layer is present, and that liquid water layer is known as whey. అంటే ఏంటి, పెరుగు మీద ఉండిపోయే ఆ నీళ్ళ లాంటివి ఏవైతే ఉంటాయో వాటిని వే అంటారు. Why? Because it is very important. It consists of carbohydrates, proteins and minerals are present, along with vitamins are present in the whey. For that reason, when it is present in the whey, this whey helps, it is a liquid byproduct of your dairy industry. First one. Second one, it consists of carbohydrates, proteins, minerals and vitamins. All these are present in this whey. First, second thing. Third one, this whey is useful in the production of antibiotic penicillin. Penicillin antibiotic is produced with the help of this whey. It is also useful in the production of vitamin B12, which is produced in the form of the vegetarians they won't get vitamin B12, because this vitamin B12 is useful for the vegetarians in the form of tablets only. For non-vegetarians it is easily available, but for vegetarians it is not that much easy to get vitamin B12, because vitamin B12 is not naturally present in the vegetarian food. Now, it is useful in the production of penicillin, antibiotic penicillin, in your vitamin B12 production, in lactic acid production. So, for all these productions it is useful. So, see here, whenever in your house, if curd is, you are taking curd, don't take that tight curd, you take along with that hard curd, you take some of the water that is nothing but whey, that helps in giving us energy. It is a carbon source. Not only that, it consists of proteins, minerals as well as vitamins. This is your whey. Next, coming to the seventh one, that is your alcohols. Alcohols are also used in the fermentation medium as a carbohydrate source. What is that alcohol? When you are taking alcohol, you can take methanol or ethanol. For some of the microorganisms, they require ethanol and or methanol for their growth. See here, you can use methane or you can use methanol. When you use methanol, the methanol is less cost and this is expensive. But methanol is less expensive and ethanol is more expensive, but some of the microorganisms, their requirement is only ethanol requirement. So, you can use ethanol only for the growth of the microorganisms. Now, when you are talking about the methanol, when you are using methanol, it is toxic and ethanol is non-toxic. See here, methanol, why it is toxic? When the methanol vapors enter into our eyes, you are not drinking methanol, simply when methanol vapors enters into our eyes, we lose our eyesight. That means loss of eyesight takes place when methanol vapors enters into our eyes. Same as that, when you are having methanol, nothing will happen. Next thing, when methanol, that is, ఎక్కడైనా మనకి కల్తీ సారా తాగారు అంటారు. ఆ కల్తీ సారా తాగితే చనిపోయారు అంటే ఎట్లా చనిపోతారు? బికాస్ దే ఆర్ డ్రింకింగ్ మిథనాల్ ఇన్స్టెడ్ ఆఫ్ ఇథనాల్. ది వైన్ ఆర్ సంథింగ్ దే ఆర్ డ్రింకింగ్, ఈ వైన్ షాప్స్లో కొనుక్కునేది ఎంత ఏంటి అంటే ఇట్ ఈజ్ ఇథనాల్. బట్ ఊళ్ళల్లో తయారు చేసే ఆ గుడుంబ వాటిలో ఒక్కొక్కసారి కంటామినేషన్ అయిపోయి మిథనాల్ ఈజ్ ప్రొడ్యూస్డ్, అండ్ వెన్ దే డ్రింక్ మిథనాల్, బిఫోర్ దే డై, the person die because of consuming that methanol, first his eyesight will be lost. That means he will first lose his eyesight, then only he will die. We know that his death has occurred, but we are not interested in knowing that whether his eyesight whether he has lost his eyesight before his death or not. We are not concentrating on that. But according to science, when a person is drinking methanol, certainly he will die, but before his death he will lose his eyesight, because the vapors of ethanol are that much toxic. So methanol is not generally used, though it is less expensive, it is not generally used as a carbon source. But ethanol is expensive, but some of the specific microorganisms, they require ethanol as a carbohydrate source, so we have to certainly use ethanol for the production of the required concentration of our target protein product. Coming to the 8th one, that is the last one, that is fats and oils. Fats and oils means you can use animal based fats, next plant based oils, fish oil. These can be used in your culture medium as a carbon source. So this oil which is extracted from fish, that is cod liver oil, it consists of a large amount of your carbon source. It consists of a large amount of carbon source when compared to the plant, and the animal fat also consists of a large amount of carbohydrate source when compared to the plant source. But the fish oil, that is cod liver oil, consists of a large amount of carbohydrate source along with the vitamins and proteins. So we prefer adding cod liver oil into the fermentation unit. Okay, now see here, then what is the use of carbon source? Why we can use any of these? I have told you 8 carbon sources. 8 చెప్పాను మీకు carbon sources: molasses, malt extract, starch, sulfate waste liquor, cellulose, next whey, alcohols, next is fats and oils. These are the 8 types of carbon sources that are inexpensive and that can be used in the fermentation medium as a carbon source. You can use any one of them according to the requirement of the microorganism. For the growth of microorganism, the microorganism requires some chemicals, and any of these can be used. అన్నీ వాడాలని లేదు, ఏదైనా ఒకటి వాడచ్చు. నెక్స్ట్, వాట్ ఈస్ ది యూజ్ ఆఫ్ కార్బన్? ఎందుకు ఇంత స్పెషల్గా చెప్తున్నా కార్బన్ గురించి అంటే, ఇట్ విల్ గివ్ ఎనర్జీ. ఫస్ట్ వన్, ఇట్ గివ్స్ ఎనర్జీ. ఫస్ట్ వన్. సెకండ్ వన్, ఇట్ హెల్ప్స్ ఇన్ రిప్రొడక్షన్. ఓకే, it helps in reproduction of the microorganism. Next, it also helps in cell maintenance. See here, it gives energy to the cell, it helps in reproduction, it helps in the maintenance of the cell. Okay, these are the functions of your carbon source. So this formulation of medium is a long question. Carbon source is a long question or short question. Okay, the carbon source is a long question or short question. Along with carbon source, this carbon and nitrogen sources, these two are used as a long question. Can be asked, that is formulation of medium can be asked as long question. Carbon source and nitrogen source put together, they can be asked as long question. Carbon source itself is a long question, or carbon and nitrogen sources both are short answer questions. So we will be stopping here and tomorrow's class we will continue with our nitrogen source. Okay students, thank you.